So today we're going to talk about antigen presentation by MHC. Lots of good stuff to talk about with this. So just as a reminder, last time we were talking about the structure of class 1 and class 2. We got all the way through talking about the structure of class 1. We didn't quite finish the details with the structure of class 2. So remember that MHC class 1 has the MHC class 1 heavy chain with 3 different domains, 2 of which come together to make the peptide binding cleft. We also have that one transmembrane domain, and we have this non-covalently associated protein beta-2 microglobulin that pairs with class 1 heavy chain to make the full class 1 complex. And these proteins really only all fold together correctly when there is a peptide in the peptide binding cleft. Beta-2M, because it's non-covalently interacting, it actually can sort of come on and off. And so MHC class 1 almost like catches and releases its peptide because the whole thing is sort of coming apart and together, like breathing. And we talked about sort of the details of anchor residues, the really important parts of binding R groups, the 8 to 10 amino acid cleft with the closed ends, all that kind of stuff last time. And then we were talking about class 2 last time. You can see that MHC class 2 is made of 2 proteins that are about the same size. Usually they're generally referred to as the alpha and the beta chain. They come together to make the peptide binding cleft, and there are 2 transmembrane domains, one on either of the proteins. Here again you can see the sort of more structural model of MHC class 2, which we talked about last time. And what I kind of left you off with is this idea that one of the key differences with MHC class 2 is that the binding cleft has open ends instead of closed ends. So you can see the peptide goes all the way across in class 2. And in fact, it can often hang off the ends. A lot of times, if it is hanging off the ends, proteases will trim the extra amino acids. But you could imagine it getting put in hanging off the ends because we have these open ends to our peptide binding cleft. These 2 alpha helices that are coming together to make up that peptide binding cleft don't have the closed tight ends at the end. As a result, the MHC class 2 peptide binding cleft can accommodate longer peptides. So with class 1, it's always 8, 9, or 10, depending on the class 1. With class 2, it's, I hear like 14 to 20, 12, like you hear sort of numbers like that. Bigger than 8 to 10, pretty big range. In addition, remember that the MHC class 2 molecule is making its interactions with atoms of the backbone of the peptide, or the main chain atoms as I have listed here. Basically that NCC, 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 that is the same from peptide to peptide. That's really what we're seeing interactions with, not with the R groups. And so MHC class 2 is much less specific in terms of the peptides that it will bind. And so there really aren't anchor residues in MHC class 2. We're not going to be able to pick out a peptide easily from some kind of peptide sequence and say, oh, that's the one that's going to bind class 2, because there's so much variation in size and we don't really have specific amino acids that are important. Sometimes people will like try to identify them and it's weak at best. And so we really don't see sort of the classic anchor residues for MHC class 2. When we talked about MHC class 1 last time, one of the things that we talked about was where MHC class 1 gets its peptides in the cell. And I told you that MHC class 1 gets its peptides from the cytoplasm of the cell. And if you remember, that was important because oftentimes a peptide in the cytoplasm might be one from a virus. And so there was this link between, well, we have MHC class 1, it's getting cytoplasmic peptides, it was going to turn on the CD8 T cells, and those are the killer T cells, which are the ones you want for a virus. So we sort of had all these pieces that linked together. With MHC class 2, the peptides are coming from a different place. We're going to see more about this place a little bit later today, as well as on Friday. So I'm going to mention it now, but you're going to be hearing more about it, which is known as the endocytic pathway. The endocytic pathway can be described in a few different ways. I actually don't know why I never asked Dr. Dunaway how he talks about it in Bio 250, so I'm going to give you a couple names for it that you'll see different names in different literature. Sometimes people call it the biosynthetic secretory pathway. Sometimes people call it the secretory pathway. Sometimes people call it the endocytic pathway. In any case, the idea right now is that we have some compartments in the cell, like the phagosome, like an endosome, or these other membrane-bound compartments that are, again, called the endocytic pathway, and we're going to be saying so much more about. And the key here is that MHC class 2 is presenting peptides that are found in the endocytic pathway. And as you'll see that when we talk about this a little bit more today, a key thing about the endocytic pathway is it's basically some kind of compartment in the cell that has a membrane between it and the cytoplasm. So there's some kind of membrane-bound compartment in the cell, and that's where the MHC class 2 molecule is getting its peptides from. Oftentimes, this might be a phagosome, like you can see at the top with a macrophage, where we might have this compartment that's created by phagocytosis, and the MHC class 2 is going to get peptides that may come in from phagocytosis. And what you can notice with this image at the top is that we have this little red pill-shaped thing, which is really kind of a bacteria that perhaps is living outside of cells normally and getting endocytosed. And so oftentimes we're thinking about things that are maybe extracellular things that are getting phagocytosed or endocytosed, being in this pathway and being potential things to be presented on class 2. Yep, Jazz.
Sorry, so you said that for MHC class 2, they're getting the peptides from the membrane between compartment and...
So no, they're getting it from the compartment.
Got it.
And so the thing that's key about the compartment is that the compartment is not touching the cytoplasm. There's a membrane in between them. And you will see more of that in some later slides today. So very often when we're thinking about class 2, we might think about, say, a bacteria that was living outside of the cell that got phagocytosed, perhaps by a macrophage, as is shown here on the top. If you recall, when we talked about class 1, I told you that the binding between class 1 and the T cell receptor was kind of weak, and we needed a partner protein to help out. That partner protein, in the case of class 1, is CD8 that was on the CD8 T cell. In the case of class 2, that partner protein is CD4. So there is this protein that's on the T cell, shown here in blue, called CD4. It also binds to the MHC molecule to a sort of conserved part that doesn't vary from peptide to peptide or MHC to MHC, while the T cell receptor is actually looking at the variable part. And so you can see we've got CD4 binding to MHC class 2, just like we had CD8 binding to MHC class 1. And a student taught me to remember this because 8 times 1 is 8 and 4 times 2 is 8, was how that they had remembered it. And The CD4 T cells, of course, are one of our types of T cells. You can kind of see this general idea here of what we've been talking about. We've got these vesicles, we've got these compartments where we're getting our peptide that we're going to present to class 2, which present on class 2, and we're going to use that to turn on a CD4 T cell. And the CD4 T cells, if you remember, are the helper T cells. They're the ones who make cytokines to help out other cells. They don't kill other cells. So if we go back for a second, here we can see that helper T cell making a cytokine to help this macrophage be better at killing things it has phagocytosed. Or we might have that helper T cell making cytokines to help a B cell better make antibodies. So those basically the helper T cell, the CD4 T cell, is going to make some cytokines that will help other parts of the response. The one other thing that we talked about when we talked about MHC class 1 that was really important was which cells express MHC class 1. So we talked about the fact that all nucleated cells express MHC class 1. So that means everything but red blood cells will transcribe and translate all the MHC class 1s. So I could pick any cell of your body, except for red blood cells, but I could pick any cell of your body I wanted, and I could find all 6 of your MHC class 1 molecules on the surface. Your A from mom, your A from dad, your B from mom, your B from dad, your C from mom, your C from dad. On the surface, any old cell I pick. Because really, any of them could be infected with a virus. Any of them could need to show off a viral peptide. And so 24/7, every cell of your body has all 6 MHC class 1 molecules on the surface and is presenting. This is not the case for MHC class 2. Only special cells can make MHC class 2. So only some cells will express, so transcribe and translate MHC class 2 and be able to do the MHC class 2 presentation pathway. I want to point out something really quickly that I'm going to write on the board just as an interesting thing. So we talked about this a little bit earlier in the semester. I told you that if we're being sort of strict about real phagocytosis, with all the sort of cell biological definitions, there are only 3 kinds of cells in your body that can do phagocytosis. They are the neutrophil, a macrophage, and a dendritic cell. And these really are both types of monocytes, so they're kind of closely related. So these are the types of cells that can do phagocytosis. If you notice, when I've been telling you about MHC class 2, I've talked about these compartments. We saw macrophage as an example for phagocytosis. There are 3 cell types in your body that can do MHC class 2 presentation. Actually, I'm going to say 3 asterisk. I'm going to mention 3 today. They're really going to be the big 3 we're going to talk about today. But I will tell you there is a 4th one that will be important later. And so the 3 important ones that I usually think about are the ones that are listed here as the professional antigen presenting cells. So those are the cells that are able to do MHC class 2 presentation. Dendritic cells, macrophages, and B cells. And you can see, for example, this figure from your textbook has like a thing about T cells that is like super in the weeds details that I wish it didn't have that, because like we are so not going there with that level of detail. But you can really see that it's B cells, macrophages, and dendritic cells. So what do you notice compared to what I've written on the board? Yeah, Emily. So neutrophils are phagocytic cells, but they don't do MHC class 2 presentation. So remember I kind of was insulting neutrophils a bit when we were talking earlier, kind of saying neutrophils can't do all the good stuff. This is part of the reason why neutrophils can't do all the good stuff. This is one of the things neutrophils can't do. However, neutrophils still have nuclei. Neutrophils can do class 1, as every other cell of your body can do. It's just neutrophils can't do class 2. Dendritic cells, macrophages, and B cells can do both class 1 and class 2. So they have like 12 on their surface. Everybody else only has 6. So one thing that you notice here is the difference with neutrophils. There's one other thing that you should notice if you compare my list of phagocytes and my list of professional antigen presenting cells here. Yep, Paige. Yeah, so there's also something interesting here about B cells. B cells are not phagocytes, yet B cells are professional antigen presenting cells. And we're not going to get into all those details today, but we will on Friday. The one other thing I want to mention, let me mention this before I forget, Jazz. The one other cell type I want to tell you about in terms of MHC class 2 presentation is the cell that's listed here, which is the thymic epithelial cell. Sometimes I think of it as a TEC, T-E-C. Basically, this is a structural cell of the thymus. The thymus is the place where T cell development is going to happen, and we need a little bit of class 2 to let T cell development happen correctly. And so the thymus does have some cells with class 2 to help with T cell development, but it's not key for like responding to pathogens or anything. It's just for T cell development. So yes, when we talk about T cell development, there is some class 2 in the thymus. It's on the thymic epithelial cells. But for when I want you to think about class 2, when I'm asking you questions about class 2, I'm asking you about professional antigen presenting cells, which are the 3 types that are shown here. Jazz, you had a question.
What's the distinction again between professional and non-professional?
So the professional antigen presenting cells, I would say, are really the ones that are presenting kind of classically through MHC, doing sort of the traditional MHC stuff that class 2 stuff we're going to talk about, are really the ones that are responsible for a good response to pathogens. The other ones can be a little more in the weeds, specialized information. So professional is really what I want you to think about, okay? Yeah. So again, if I looked at any cell of your body, it would have the class 1 molecules on the surface, but not the class 2. But if I looked at a dendritic cell, B cell, or macrophage, it would have both all the class 1s and all the class 2s codominantly expressed on that cell and presenting on that cell. So this slide and the next slide are really just sort of comparing contrasts of class 1 and class 2. So you can see we can compare which domains of the protein make up the peptide binding cleft. You can see for class 1, it's 2 parts of the alpha, the main heavy chain protein, alpha 1 and alpha 2. For class 2, it's something from each different chain, alpha 1 and beta 1. Class 1 is closed at both ends, class 2 is open at both ends. We see differences in the length of the peptide. You can see sort of anchor residues being really key for class 1. And so you can kind of see these differences. If we look at a similar table, you can see the difference in the structure. You've got this one alpha chain for class 1 that pairs with beta 2 microglobulin, where you have varying alpha and beta chains for class 2. Class 1's on all nucleated cells. Class 2 is really on our professional antigen presenting cells. Class 1 presents to CD8s. Class 2 presents to CD4s. Class 1 has cytosolic proteins. Class 2 does endosomal proteins. So this is sort of the compare and contrast we're going to talk about. But what we haven't really gotten into yet is this sort of earlier part of the MHC process. So how we go from some protein in the cell to having a peptide from that protein on MHC class 1 and the MHC class 1 on the surface of the cell. Today we're going to talk through the steps of this process for MHC class 1. We'll do MHC class 2 on Friday. So I'm going to focus on the class 1 parts of it today. And in order to kind of talk about this, I have to give you, again, a little bit of cell biology, just to make sure we're all on the same page of some details of cell biology. So this is, in a lot of ways, how I, as an immunologist, can sometimes view the cell. And actually, I'm not even really caring about the nucleus in this situation. But what you can notice is that I have the area outside of the cell, which is in yellow. And I have the cytoplasm, which is in light blue. And I also have some compartments inside the cell. And you'll notice all those compartments are also shown in yellow. Basically, all the yellow stuff is across a membrane from the cytoplasm. So everything in yellow is not touching the cytoplasm. If you wanted something to go from a yellow place to a light blue place, you have to cross a membrane. And if you remember from Bio 250, crossing a membrane takes energy, takes effort. You don't just get across a membrane really easily. And so for me, I often think about, are you in the cytoplasm or are you across the membrane from a cytoplasm? In the sort of light blue, or are you in the yellow? What you can notice is that if something gets brought into a cell, here it's shown as endocytosis, but it would be the same thing as phagocytosis. Basically, we're taking a little pinch of the area outside of the cell and we're making it a compartment. And so in a lot of ways, that compartment has a lot of similarities to outside of the cell. They're both across a membrane from the cytoplasm. And if we're going to secrete things and send them outside of cells, that has to go through this pathway of vesicles to get secreted outside of the cell. And so I often kind of think about the cell as divided into two zones. The cytoplasm and the across a membrane from the cytoplasm. Or the it would require me work. Or it would require me energy to put something there from the cytoplasm. All of those parts of the cell that are shown here in yellow, all of those vesicles where we are bringing things in, like the endosomes, like the lysosome, or all of the vesicles and compartments where we're bringing things out, like the ER or the Golgi, or all of these other types of vesicles, you can see are sort of connected to each other by arrows. So stuff can sort of move between those different compartments by little vesicles blebbing off from one and fusing with the other. And you'll see that on the next slide. And so this whole interconnected area of these yellow compartments is this biosynthetic secretory pathway, also known as the endocytic pathway, also known as the secretory pathway. And so again, we're often going to be thinking, as we're thinking about class 1, as either in the cytosol or in the secretory pathway. And those are the two choices that we can have. And so here you can see kind of this other key way of looking at this same process of intracellular trafficking of proteins. When the cell makes a protein, when we do translation, there has to be a choice of, is this a protein that's supposed to be in the cytoplasm, or is this a protein that's supposed to be in the secretory pathway? Basically, is this protein supposed to be in the cytoplasm, or does this protein have to be across a membrane from the cytoplasm? Or does this protein have to be in a membrane? Because either in a membrane or across a membrane requires effort. The ribosome is chilling in the cytoplasm. Here you can see the ribosome in the cytoplasm. It's making the protein in the cytoplasm. If we want something either embedded in a membrane, or we want to secrete something outside of the cell, or we want to put something in any one of these locations, we've got to use some energy to do that. The way that that works is actually the ribosome can tell when it's translating the protein where that protein's supposed to go. I know, super cool cell biology. And the ribosome, if it needs to have that protein go across a membrane or into a membrane, the ribosome basically goes to the membrane and translates there and uses the actual physical force of translation and of the amino acid coming off the ribosome to get a push across the membrane, sort of taking advantage of that push. That's what's happening at the rough ER. That's why you sometimes have some translation happening at the rough ER, some ribosomes on the ER, because they're like, I found an mRNA that needs to make a protein that needs to be secreted or transmembrane or whatever. So every transmembrane protein in your body has to get translated this way. Any secreted protein has to get translated this way. And so basically there's this big step of you're either in the cytoplasm or you go somewhere else. Yes, there's other somewhere else's. I don't care about them right now. You can be in the cytoplasm, or you can get translated into the ER. If you get translated into the ER, then you can sort of move around from the ER to the Golgi, to the vesicle, to the lysosome, or to the cell exterior through this process. One reason why this is important to think about is that MHC class 1 itself is a protein that is going to be displayed on the cell surface. So MHC class 1 itself has to be translated at the ER. MHC class 1 itself is translated at the ER and put in the membrane at the ER. And that eventually gets sent up onto the cell surface. So this tells us a little bit about biosynthesis of class 1, but it's going to deal with another really key problem that we're going to see. And so this is the other thing I want to tell you about kind of the biosynthetic secretory pathway. Really, we're talking about something called topology of transport. In the left, you can see a bilayer of lipids. And I know it's a bilayer because it's got two layers, a blue one and a green one. So I can see blue lipid and green lipid. And what you might notice is that the blue lipid is touching the cytoplasm, and the green lipid is touching the inside of the compartment. And you can also see the way that we move things between different parts of the biosynthetic secretory pathway. So what will happen is we will have a little vesicle bud off of one compartment and then eventually fuse with the other compartment. So basically we're carrying stuff from one compartment to another. What I want you to notice in this process, this is also true in the image on the right, where here it's a orange lipid is touching the cytoplasm and blue is touching not the cytoplasm, but it's the same idea, is that the stuff that's touching the cytoplasm, the stuff that's on the cytoplasm side, always stays on the cytoplasm side. And the stuff that's not on the away from the cytoplasm side always stays away from the cytoplasm. So what you can notice is when we bud off and make this transport vesicle, you can see that we had blue on the outside, blue lipids on the outside. It buds off and blue stays on the outside. And then when we merge this vesicle into the target vesicle, the blue stays on the outside. The green always stayed on the inside. The green was never touching the cytoplasm. Similarly, the pink dots were away from the cytoplasm, and they stay away from the cytoplasm. They're never going to touch the cytoplasm again. Once they get into a compartment, they're in the compartment forever. And they're never touching the cytoplasm again. They're always staying inside the compartment. You can also see with this transmembrane protein, the round part is towards the cytoplasm. The pointy part is towards the inside of the vesicle. When we bud off, the round part stays towards the cytoplasm. The pointy part stays towards the inside of the vesicle. And when we fuse again, we get the same kind of, we keep that same orientation. Round part in the cytoplasm, pointy part away from the cytoplasm. And you can again see this in the view on the right as well. So sort of which side of the membrane you're on at the beginning of this process matters a lot. Sort of like you choose at the very beginning at translation, is this protein on the outside of a membrane or on the inside of a membrane? And then it stays in that orientation the rest of the way, sort of moving around the cell. Flipping something over in a membrane is like energetically, catastrophically hard. This also is true at the cell membrane. So we can see the Golgi here, and we can have, sometimes we have vesicles that will leave the Golgi and fuse with the cell membrane. That's how we get things secreted, or that's how we get things to be transmembrane proteins. So you can see if we have something that's inside the Golgi, it would be inside the vesicle, and then when that vesicle fuses with the cell membrane, the stuff gets secreted. So if we want to secrete something, like, oh, I don't know, a cytokine or whatever we want to secrete, we have to get it inside a vesicle for it to actually get secreted correctly. If we want something to be a transmembrane protein, like this little round protein, you can see we have to make sure the round part is inside the Golgi and inside the vesicle if we want it to be outside on the surface of the cell. So it matters what direction that protein is here so that it can be in the right way at the surface of the cell. And you can see sort of this process where we have stuff in a vesicle, that vesicle fuses with the surface of the membrane, and we secrete. And if we bring something in by endocytosis or phagocytosis, it's really the same thing. We're basically wrapping a little vesicle around whatever. So these are sort of key things you have to be aware of. So I want you to think about this for just a second, okay? We're going to be thinking about putting MHC class 1 on the surface of our cell. Our goal is to have MHC class 1 on the surface of the cell, showing off the peptide, being like, here, T cell, do you see this? Do you see what I have, right? So the peptide binding cleft has to be, outside the cell, pointing away from the cell, right? Ready to show to the T cell. So when MHC class 1 is biosynthesized, where does the peptide binding cleft need to be? Yeah, Paige. Okay. Julie. So we want at the end, we want it to be outside the cell, right? So where does it have to be when it was biosynthesized? Inside where? It's inside the compartment. It's got to be inside the vesicle. The MHC class 1 binding cleft should be more like the roundy part of this protein. So we actually are going to care, even from the beginning of it, that we make it in the right direction. The key thing I want you to notice here is that the binding cleft is going to be inside this vesicle. The binding cleft is not going to be touching the cytoplasm. That's going to be really, really important here in this process. I really like cell biology and would love to tell you more, but I cannot. Because now I'm going to tell you about class 1. So the steps of MHC class 1 presenting are steps that are done in every cell type, basically all the time. And when I first was a TA for an undergrad immunology class, the person I TA'd for broke down this processing and presentation stuff into 6 steps. So I often think about it as 6 steps. So we're going to talk through the 6 steps, okay? Some of the steps you're going to hear and be like, that is a dumb step. Like, why does that get to be its own step? The reason is because when we do compare and contrast with class 2, something interesting is going to happen with class 2. So when you hear something and you're like, that is the dumbest thing that she is calling a step, there's a reason, I promise, okay? And so the 6 steps are, I know how to spell. The 6 steps are acquisition, or sort of acquisition of antigen. How do we get the antigen? How do we get the peptide? Tagging. Sometimes we can also say tagging for destruction. Then destruction. Since it's a protein we're destroying, we can call it proteolysis. Delivery. How do we get the peptide and the MHC in the same place? Binding. How do we get the peptide bound to the MHC? And then display. How do we show this completed thing on the surface of the cell? So those are the 6 steps we're going to see for class 1. And here is sort of the overall process of class 1. I am going to sometimes reference back to figure 5.17 from your textbook. There are things I both love and things I hate about figure 5.17. So we're going to see some of figure 5.17 today, but we're also going to trash talk figure 5.17 on Friday. Because this is sort of showing us a pretty nice overview of MHC class 1 presenting its cytoplasmic peptides. Sort of a general thing here, note that I told you that as an immunologist, I think of the cell as having 2 parts, the cytoplasm and the not cytoplasm. There are 2 kinds of MHC. One is going to deal with the cytoplasm, and one is going to deal with the not cytoplasm. When we talk about proteolysis, if we were in a general cell biology class, I would tell you there are 2 general ways we destroy proteins in the cells. One is in the cytoplasm, and one is in the not cytoplasm. So this divides out pretty nicely in terms of the way that we're going to think through this. For class 1, acquisition of antigen is relatively lame. For class 1, the way we acquire the antigen is that we have it in our cytoplasm. In a lot of ways, the way that I think about acquisition of antigen for class 1 is that you got infected. Basically, you don't have to try to acquire the antigen. You don't have to do anything actively to get this peptide. It's just in your cytoplasm. With class 2, you're going to have to try to get the peptide. You're going to have to do something to acquire that antigen. With class 1, it's just in the cytoplasm. You're getting infected. So it's sort of like being infected or just being in the cytoplasm. So like I said, pretty lame. Now we need to think about how we're going to tag this protein to get destroyed and how we're going to destroy it. There are two general ways, like I said, that we can destroy proteins in the cell, and this is going to be one of the two of them. And I want you to understand about this process of degradation that sometimes we degrade a protein because there's a problem with it. Perhaps it's misfolded, perhaps it has some sort of issue, perhaps it's been modified in some way. There's something bad about that protein. But sometimes we degrade a protein because we want a new one, like with a phone. Sometimes there's nothing wrong with your old phone, but you get rid of it to get a new one. Basically, every protein in the cell has like a general half-life, where it just after a while gets degraded and you make a new one. And in some ways that's like a quality control thing, like maybe the protein got messed with and you can't detect it, so you just degrade them after a while. But what I want you to realize is that this process that I'm going to be telling you about in terms of proteolysis is happening basically to every protein in the cell at some rate. Your cell is like ditching its old proteins and making fresh ones at some rate all the time. So nothing I'm telling you here is necessarily specific to a protein that's coming from a microbe. This could happen with any protein that is in the cytoplasm. And the thing that can happen is that we can actually add a tag onto this protein. So you can think of it as adding like a little sticky note. It is a covalent modification. So sometimes when we're going to talk about different things, they're going to be non-covalent modifications, but this is in fact a covalent modification. We are actually putting this tag covalently onto part of the R group of a lysine. And this tag is called ubiquitin. Sometimes people refer to the process of adding ubiquitin to things as ubiquitination. Other times people talk about this process as ubiquitination. They are both the same thing. Those two words mean exactly the same thing. And so the basic idea is you're going to use a set of enzymes in order to add this tag, this ubiquitin tag, to some protein that you want to degrade. And this is happening to all cells at some rate, or all proteins at some rate in the cells. Ubiquitin itself is a protein. It is a very small protein, 8.5 kilodaltons. It's this little tiny protein that's getting stuck onto other proteins. Again, like a little sticky note. And so you can see this process happening here. If I was doing kind of classic cell biology, I would tell you that adding ubiquitin to a protein can give you multiple different outcomes. The outcomes are different based on how many ubiquitins you add and what kind of biochemical linkage you use to add them. In the situation I'm telling you about here, we're adding a string of 4 ubiquitins. So we're adding 4 ubiquitins in a line like this. They're going on a particular lysine. They have a particular kind of biochemical linkage. There are situations where ubiquitin does something else if it's bound in a different way or not in a chain of 4. So here we are technically seeing a chain of 4 ubiquitins. This is the thing you've learned about if you've learned about ubiquitin before. And so when we have added ubiquitin, 4 ubiquitins to some protein, that is a signal to the cell, that is a marker that the cell should destroy that protein. And we're going to use one of our 2 cellular trash cans, because we got 2 cell trash cans that allow us to degrade stuff. Oh my gosh, one's for the cytoplasm and one's for the not cytoplasm. And our thing that we're going to use here is called the proteasome. And so we're going to tag things to be destroyed by putting ubiquitin on them. And then we're going to destroy them using the proteasome. So the proteasome is an organelle in the cytoplasm. How do you know it's an organelle? From just what's on this slide, how do you know it's an organelle? Don't overthink it. How do you know it's an organelle? If it ends in -ome, it's an organelle. So it's an organelle. And what kind of organelle is it? It's a protease organelle, a proteasome. So the idea with the proteasome, or the proteasome, is that it's an organelle. It is basically just an organelle that just breaks up proteins, that does proteolysis. So it's this special organelle that is going to do proteolysis of anything that has that ubiquitin marker on it. Basically, if there's that ubiquitin marker, we're going to take that material and we're going to send it to the proteasome. And you can see that at the end, we're going to get out some peptides. I have to tell you a little bit about the structure of the proteasome and sort of exactly how it works. And so here you can see other examples of the proteasome. If you look in this previous figure, you can see it sort of shows the proteasome as having like little rings of purple circles and then a blue thing on either end. And that is kind of accurate in that there are these rings that are made of individual subunits. So we have these rings that make up the core. You can see, this is the view from your textbook, and you can see these ringed proteins that make basically a little chamber in here to do proteolysis. And so again, you can see the proteasome is shown looking kind of like a little barrel, where you have these different little proteins making up rings of the barrel and giving you this inside chamber to the barrel. And that inside chamber is where proteolysis happens. That's actually where we're going to cut stuff up. That's where the protease activity actually is. And then you can also see that, and there's 4 rings, each made of a few different subunits. You can also see we've got a nice cap, the cap on either end. That's important because it's going to actually recognize the ubiquitin and make sure only stuff with ubiquitin on it gets destroyed. And it's going to, keep rando stuff from going in and getting destroyed by having this nice cap on either end. So this is really what the proteasome looks like. And you can see sort of the general idea of proteasome recognition here. So basically we've got the cap. The cap is going to recognize the ubiquitin, and then it's just going to start shoving the protein into the barrel part, where we're going to get degradation down into peptides. And so you can see it recognizes the ubiquitin and just sort of unfolds the protein, sort of strings it into the chamber part where we get proteolysis and we get peptides out. The nice thing about the cap is the cap also takes the ubiquitins off and recycles them so that they can get put on other things that need to get degraded. And so you can see here's my ubiquitins on my protein. The cap's going to recognize. We're going to start shoving the protein into this barrel, we're going to get peptides out with our proteasome. So that's sort of the basic proteasome, which is really key for proteolysis in MHC class 1. In some situations, so all your cells 24/7 are doing this. In some situations, the proteasome can be modified. If, for example, a cell receives some interferon, that cell's going to be like, oh, I might have a virus. I should do really good class 1. I should be awesome at proteasoming. I want to specialize this, make it look really good. Sometimes the proteasome can get modified into something called the immunoproteasome. The basic idea with the immunoproteasome is that when cells are induced, they switch out some of the subunits in the ring. So you can see this is a regular proteasome. We can switch out some subunits in the ring. And as a result, we get slightly different peptides that are getting made by the proteasome. It just so happens those peptides are super awesome at getting on MHC class 1. So one example, there's lots of examples, I'm only going to give you one example, is that the regular proteasome makes a whole bunch of different peptides. Sometimes it makes peptides that are 2 amino acids long, and sometimes it makes 3s, and sometimes it makes 4s, and sometimes it makes 77s, and it makes a bunch. When you have the immunoproteasome, it makes 8s, 9s, and 10s. And only 8s, 9s, and 10s. Because those are the best ones for MHC class 1. So there's sort of this specialization to have a proteasome that's even better. And this is one of those things that can get turned on when we're seeing interferon production in the antiviral state. So our step of proteolysis is with the proteasome. So that was really how we made the peptides, okay? The next step is that we need to deliver the peptides to the MHC class 1 molecule. And so if we're going to talk about this issue of delivering the peptides to the MHC class 1 molecule, we need to think for a second about the MHC class 1 molecule and what it's been doing all this time. We kind of have already implied this. The MHC class 1 protein is made in the endoplasmic reticulum because it needs to be presented on the surface eventually, presented outside of the cell. It is made without a peptide in the peptide binding cleft, which means it's not actually able to fold stably because it needs a peptide to be stably folded. And so that MHC class 1 molecule is going to interact with a lot of different chaperones and just be kind of hanging out in the endoplasmic reticulum like this, with its empty peptide binding cleft, waiting for that peptide to come into the peptide binding cleft. So this is sort of where we are right now. We've got our peptide fragments made in the proteasome. We have our MHC class 1 heavy chain chilling with the chaperones, ready to receive a peptide. And this image also shows, because I modified it, the problem of MHC class 1. And so there is a key problem that comes up when we think about MHC class 1 presentation, which we can see here. So this problem has to do with delivery, getting those peptides to the MHC class 1 peptide binding cleft. What's the problem? Yeah, Roxy.
It looks like it's blocking out, closing the fragments inside.
So you mean the chaperones?
The chaperones are
So the chaperones make it a little tricky, but the chaperones are going to let it happen. We're not going to worry about the chaperones. But there's another problem. Yeah, Emily.
The peptides are in the cytoplasm, but the cleft is really not cytoplasm.
The peptides are in the cytoplasm. The open place where they need to bind is in the ER, in the not cytoplasm part of the ER. And it's actually really important that it's there because we want to get this on the cell membrane. So the peptides and the place where the peptides need to go have a membrane in between them. They're not in the same place. So this is kind of a problem in order to get binding to happen. And what was eventually discovered was that there is a specific protein called TAP. TAP stands for the transporter for antigen processing. And TAP's job is to actually shoot the peptides into the ER. And so TAP actually uses ATP to shoot peptides from the cytoplasm into the ER. So it does the delivery. The discovery of TAP was fabulous. Lots of cool stuff about the discovery of TAP. And so basically TAP is using ATP and shooting all the peptides from the cytoplasm into the ER. And this is like a key step. Without this, none of this would actually work. None of this would happen. And then we can think about binding. And the thing that's actually really key with TAP as well is that one of those chaperones, it's shown here in blue, and it's called tapesin. Tapesin actually makes sure that the MHC class 1 is next to TAP. So basically, as peptides are getting shot into the ER, the open cleft is right there next to it, ready to bind the peptides. And so tapesin is basically holding everything in place. So tapesin is holding MHC class 1 and TAP right next to each other, so that as the peptides come into the ER, they can bind straight to the MHC class 1. They're not going to go some other random place, because tapesin is sort of holding everybody together and making sure that binding occurs. And so now you can see tapesin is binding, holding TAP next to class 1. Now we can get our peptides onto class 1. And basically, if the peptide has the right anchor residues, it binds. And it binds with good affinity. So we just get binding through kind of good biochemical principles if we have good anchor residues. Once that process happens, once we get that binding event to occur, basically the chaperones release the MHC class 1 molecule with its peptide. And that MHC class 1 molecule with its peptide is able to just continue through the secretory pathway to get put on the surface of the cell. I feel like maybe I've said this word here to you guys before. I don't remember if I've defined this general term or not. But the term for what's going on here is actually that this process is constitutive, which means automatic. Doesn't require any kind of extra signal to make it happen. So basically, once the binding's there, once we're all set and ready to go, once that MHC is ready and is released from the chaperones, it just automatically goes onto the cell surface. It's constitutively displayed. And you can see that, display happening here. So basically, here you can see our proteasome making peptides. In this figure, one of the things I dislike about the figure is that we have magic happening to put the peptide in the ER. We get binding, and then as soon as we get binding, we just display that at the cell surface constitutively through sort of normal, old, like average biochemical cell or cell biological processes. Cell doesn't have to try. So this is sort of our overview of the MHC class 1 presentation pathway. and you're going to see some additional overviews in a little bit. But there's one other part of MHC Class 1 presentation that I want to sort of mention to you a little bit here. And it's something that's going to come up later and be so important in this class. And I also joke, because I really like this image from this review, but every time I show it, Dr. Dunaway yells at me and says there's parts of it that are inappropriate. And so I have like modified it so heavily to try to take out everything he doesn't like, but still use it. But it's a ridiculous image at this point with all my modifications. So what I want you to realize is that if we're starting with some kind of protein, we're not necessarily going to present that whole protein. We're just presenting a peptide that came from that protein. So you can see the proteasome is going to cut it up into different kinds of pieces. And those pieces may get put onto MHC Class 1 and then presented on the cell membrane so that T cells can check out that cell and see if this is happening. And what you want to notice, this is the one that he has so many issues with and I have modified so well. What you want to notice is that our protein is probably getting cut into many different pieces. It's not as if the protein is always getting cut at amino acid number 7. Every copy of the protein is getting cut in slightly different ways. We're making slightly different pieces. And all of those different pieces are ending up in the ER, and they may bind to different MHC proteins, which can potentially present different ones to the CTL. So the key reason why we have all of these different MHC molecules is so we can be presenting different parts of our peptide. And so any peptide, so we're not generally presenting, one peptide from any particular protein antigen. We're actually usually presenting many epitopes that bind to our many different MHC Class 1s. And so you can see our cell has many different MHC Class 1s on its surface. What I also want you to realize is that this starts with a protein that is in the cytoplasm. There are tons of proteins in the cytoplasm. Your cytoplasm's full of proteins. All your proteins, all your cytoplasmic proteins are in the cytoplasm. Thank you, Captain Obvious. You are degrading them at some rate, like the iPhone. And so in fact, all of your cells 24/7 are presenting self-peptides on MHC. Nothing I've told you here is specific to a viral peptide or a pathogen peptide. Basically, every protein in the cell is getting cut up. Every protein in the cell is getting shown on the surface on MHC Class 1 to T cells. Self-peptides all the time. And so here you can see, there are self-peptides and potentially foreign peptides on the surface of any infected cell. Each cell has multiple copies of MHC, maybe some self-peptides, some foreign peptides on each. So self-peptide is always, always, always being presented on all of your cells all the time. In the case of Class 1. And so you might say, why do I not have all sorts of CD8 T cell autoimmunity? Why am I not making all sorts of CD8 responses against self-proteins? And the answer is because it's something that happens on the T cell side, the T cell development side, not on the presentation side. The cell doesn't know this is a good peptide or this is a bad peptide. It just presents everything on the cell surface and leaves it to the T cell to know if that thing is good or bad. So self-proteins are being presented all the time.